So receptor potential is also known as the graded potential. It is so because the size or the magnitude, its magnitude or the size depends on or varies depending upon the strength of the stimulus. It's the membrane potential of the receptor, mainly the sensory receptors, which triggers the action potential. It requires the stimulus to bring the resting membrane potential to either the threshold or to more negative potential, depending on the stimulus, whether it is excitatory or inhibitory. The channels involved include the ligand-gated channels. So let us understand it more seeing the figure. So as I said, the channels involved are ligand-gated channels. So in case of excitatory one, when the stimulus comes to the presynaptic neuron, glutamate, which is excitatory neurotransmitter, is released from presynaptic neuron, which will go down to the postsynaptic neuron, and it binds to the sodium and calcium gated channels, which will cause the influx of sodium and calcium, which would lead to the changing of polarization of the membrane. So the membrane here, in case of excitatory stimulus, will get depolarized. On the other hand, to the inhibitory one, when the stimulus comes, GABA, which is inhibitory neurotransmitter, will be released from presynaptic neuron, and it again goes and binds to the postsynaptic neuron to the chloride-gated channels. So the influx of chloride will take place and simultaneous influx of sodium and calcium takes place, due to which again the membrane is polarized, but this time it will be hyperpolarized. So this was about receptor potential. Now again this is the graph wherein the half of the right part shows where the depolarization of the membrane occurs due to graded potential, and the left half shows where the hyperpolarization occurs, which is due to receptor potential. Now moving on to the action potential. When the stimulus comes, the resting membrane or the resting state, when the membrane is in resting state, it will go to threshold membrane to threshold potential to minus 55 millivolts, from where the depolarization occurs, which is due to the influx of sodium, followed by repolarization due to efflux of K+, and then the hyperpolarization state is arrived due to the slower closure of the potassium, and again the resting potential is achieved. What my topic was about comparing and contrasting, which means both similarities and differences should be discussed. So concluding this both, the similarities which I came to know about were first, both involves the movement of ions. On the product concept, I mentioned it here ions. It can be sodium, potassium, any of them, which would cause depolarization, and in cases it even causes hyperpolarization. Both are triggered by the stimuli. Stimuli can be in any form like light, touch, chemicals, and they will convert them into electrical signals. The stronger stimuli, the third point is important point in this, as it mentions, stronger the stimuli, it will generate larger receptor potential, which would be easier for the membrane to reach from resting membrane potential to the threshold, which will further be easier to achieve the action potential. Now, moving on to the differences. Based on location, graded potential, it is majorly seen in dendrites, cell body, and mainly the sensory receptor. Whereas in action potential, axon or majorly the axon hillock, it is seen. Types of channels involved in graded potential, they are ligand-gated channels. Initially, ligand-gated channels are present when the membrane is depolarized enough. Then the voltage-gated channels opens, which causes action potential further. But in action potential, only voltage-gated channels are involved. The ions involved briefly in graded potential are usually sodium, potassium, and chloride. But in action potential, majorly sodium and potassium ions are seen. Summation, this is the main point where in graded potential, it can be summated. What does summation means is actually giving multiple times the stimulus to the membrane so that it would be easier to reach the threshold. In receptor, generally the membrane is unable to reach threshold, but in case of summation it is possible. But in action potential it cannot be summated. The now main or the major three differences. Graded potential, it does not obey all or none law, but action potential does. In graded potential, it is non-propagated, that is, it dies down over shorter distances, but in action potential, it is propagated. In graded potential, as I told earlier, stimulus does not reach the threshold level. In action potential, stimulus reaches the threshold, thereby it causes action potential. Now, this figure is the overall view of the location of receptor and action potential, as receptor is in the dendrites and cell body, but the action potential goes on to lobular distance.